I'm Dr. Fakir Averi, and this is Bacteremia in adults. Here are some reliable resources for this topic. Infectious Diseases Society of America, or RDSA, provides guidelines, and we will focus on IDSA guidelines for bacteremia. First, in 2009, IDSA released guidelines for the diagnosis and management of intravascular catheter-related infections. Then in 2011, IDSA had specific guidelines for MRSA, or methicillin-resistant Staphylococcus aureus infections. This is a wide variety of infections due to MRSA, including bacteremia. So there is a section in there that's specific to bacteremia due to MRSA. There is also a consensus guidance that was published in 2020-21 for optimizing the management of uncomplicated gram-negative bloodstream infections. And more recently, IDSA has a guidance on the treatment of antimicrobial resistant gram-negative infections. The next learning objective is describe the common mechanisms of bacterial resistance to antibiotics. Wild-type bacteria are susceptible to many antibiotics, and they are called wild-type because they have not developed resistance through mutations or acquisition. However, they can still be resistant to some antibiotics intrinsically, and that's what determines the spectrum of activity of antibiotics. Resistant bacteria are the ones that are resistant to antibiotics that normally should have been susceptible to as wild-type. For example, wild-type Staphylococcus aureus is susceptible to penicillin, but you will see that many Staphylococcus aureus strains have developed resistance to penicillin. Wild-type Pseudomonas aeruginosa is intrinsically resistant to penicillin. Now, when bacteria develop resistance, not only can they pass these resistance genes when they divide, but they can also pass it to other species. The transfer of resistant genes occurs generally through 4 different mechanisms. Transformation is when naked DNA transfers from dying bacteria to a competent recipient. Transduction is the packaging of genes, usually from small plasmids, by transducing bacteriophages. One characteristic of bacteria is that in addition to their chromosome, they also have DNA as a circular DNA, which is referred to as plasmid. And this plasmid, because it's small in general compared to chromosome, it can easily be transferred to other bacteria. In conjugation, which is the most efficient way of transferring resistant genes to other species, self-transferable plasmids mediate direct contact by forming a mating bridge between cells. You can see in the picture in the right-hand side the different steps of conjugation. Lastly, there can be conjugative transposition. You see, as opposed to chromosomes and plasmids, transposons are specialized sequences of DNA that possess their own recombination enzymes, allowing transposition or hopping from one location to another, independent of the recombinant enzymes of the host. Here are the common mechanisms of antibiotic resistance. Remember, for antibiotics to work, they need to get to the site of action. This provides several opportunities for the bacteria to prevent antibiotics from getting to the site of action. The first mechanism is reduced permeability. In this picture, we're looking at gram-negative bacteria with 2 membranes, so there's the inner and outer membrane. And for many antibiotics to get to the site of action, they need to penetrate through the outer membrane, often by going through a Porin channel. So one thing that bacteria can do is to reduce the production of these Porin channels, or in some cases even loss of Porin production. And that will basically eliminate the channel of entry for antibiotics. There is also something referred to as biofilm, which we will discuss when we talk about bone and joint infections. Now, if the antibiotics manage to go through the outer membrane, some organisms can actually develop efflux pump and pump these antibiotics out. This is a common mechanism in Pseudomonas aeruginosa. Another mechanism is the change in antibiotic targets by mutation. So if the antibiotics do get to the site of action, then the site of action can mutate so that the antibiotic can no longer bind to the site of action. The fourth mechanism is modification of antibiotics itself. So there are beta-lactamases that can actually break down antibiotics. There are also aminoglycoside-modifying enzymes that can basically alter aminoglycosides. And lastly, there is alteration of metabolic pathways. For example, the evasion of common pathway to make folic acid, which leads to resistance to trimethoprim sulfamethoxazole. Now, there are other mechanisms of resistance, but these are the common ones that we will focus on. Now, let's compare gram-positive and gram-negative bacteria. So on the left, we can see gram-positive, which only has the inner plasma membrane. There is no outer membrane. So on top of the membrane is the peptidoglycan layer. With gram-negative, there are two layers. So there's the inner and outer membrane, and between the two membranes, there is a small layer of peptidoglycan. Beta-lactams prevent the formation of peptidoglycan. So their site of action is where peptidoglycan is. So in order for the bacteria to make beta-lactamase and break down beta-lactams, they need to be made around the peptidoglycan layer. So you can see that for gram-positives, so this will be actually outside of the cell, whereas for gram-negatives, these beta-lactamases will be basically in the periplasmic space between the two membranes. This basically shows you that with gram-positives, they are likely to lose a lot of these beta-lactamases to the environment because there is nothing to hold all these beta-lactamases as they're being made by the bacteria. Whereas for gram-negative organisms, because of the outer membrane, they can hold on to beta-lactamases. So this is more cost efficient for gram-negative. So you will see that beta-lactamase production is a lot more common in gram-negatives compared to gram-positives. ESBLs are an important mechanism of resistance in gram-negative organisms. But before we can talk about ESBLs, let's consider what is a beta-lactamase. So beta-lactam ring is essentially a 4-member ring, and monobactams only have this 4-member ring, and penicillin, cephalosporins, and carbapenems have 2 rings fused together. And what some bacteria can do is that they can make a protein called beta-lactamase, which can essentially break down the beta-lactam ring, and by opening it, they will make the beta-lactam inactive, and that is a mechanism of resistance for them. And there are a lot of beta-lactamases out there. So there are penicillinases that can break down penicillins in general. And then, later on, when we brought cephalosporins to the market, Bacterial learned how to make cephalosporinases, which can break down cephalosporins. And later, when we had carbapenems on the market, they learned how to make carbapenemases. And collectively, these are all referred to as beta-lactamases. And of course, to fight that, we also made beta-lactamase inhibitors. And what beta-lactamase inhibitors are essentially compounds that look like a beta-lactam ring, so that they have affinity for these enzymes that bacteria make. And by binding to the beta-lactamase inhibitors essentially make this inactive, so that these beta-lactamases can no longer inactivate the actual beta-lactam, so the beta-lactam can remain active and kill the bacteria. And we have quite a few of them on the market now. We have clavulanic acid, tazobactam, sulbactam, avibactam, weberbactam, and more recently, relibactam. Now, what exactly are ESBLs, or extended spectrum beta-lactamases? So these are the enzymes that can hydrolyze extended spectrum cephalosporins and monobactams. And extended spectrum cephalosporins are essentially 3rd generation cephalosporins, such as ceftriaxone. And they do not hydrolyze cefamycin, so cefamycin are a subset of 2nd generation cephalosporins, and they can also not hydrolyze carbapenems. Now, ESBLs are susceptible to most beta-lactams with the exception of sulbactam. So sulbactam does not have good activity or affinity against ESBL. Now, the CDC antibiotic resistant threats in the United States document lists germs into one of three categories, either urgent, serious, or concerning. For ESBLs, the CDC has labeled these as serious threat, so this is in the middle, as they are attributed to increasing cases and significant mortality in the US. In fact, when you see year by year, the number of cases of ESBL infections has been increasing year after year. ESBL producing Enterobacterialis often cause infections in otherwise healthy people. So anybody could get these infections, and about a quarter of patients with these infections had no known underlying health conditions. Now, antibiotic options to treat ESBL producing Enterobacterialis infections are limited, and healthcare providers often need to use IV carbapenem antibiotics for infections that otherwise could have been treated with oral antibiotics. So that increases number of patients who need to be in the hospital setting. Now, there are hundreds of beta-lactamases out there. So it's really difficult to study and see what's clinically significant. So the Ambler molecular classification of beta-lactamases, which was introduced back in 1969, it's not perfect, but it's perhaps one of the simplest ways to look at beta-lactamases, because it essentially breaks them into four classes. So class A, class B, class C, and class D. Class A, which is a large class, is a bit tricky because it has several subclasses. So this includes penicillinases that break down penicillins, and cephalosporinases that will break down first and second generation cephalosporins. And these can either be genes on chromosomes or plasmid. For Enterobacterialis is most commonly on plasmids. Some examples are TEM1 and SHEV1. But what's really clinically significant is the ESBLs that we just covered. So ESBLs, and again, there are many enzymes under the subclass of ESBLs, and these are most often on plasmids, but they can also be on chromosome. But what's clinically significant is essentially the ESBLs that resides on plasmids. And in general, ESBLs can be broken into four categories of either TEM-derived. So essentially these TEM1 mutations over time have led to TEM-derived versions of ESBLs, as well as SHEV-derived. So mutations to SHEV1 over time has led to many descendants of this enzyme that are under ESBLs now. But the most common enzyme under ESBL is the CTX-M. So CTX-M is what's most common in the US and worldwide. And as part of class A, we also have carbapenemases. And before I mention this, let me go back to ESBL. So ESBLs essentially not only break down, extended spectrum cephalosporinases, so third generation cephalosporin, but they will also break down penicillins and first and second generation cephalosporins. And then when you look at carbapenemases, even though the name says it breaks down carbapenems, but these enzymes also break down penicillins and cephalosporins as well as carbapenems. So KPC is the most common enzyme in the US. Now, when it comes to class B, these are the metallo-beta-lactamases. Essentially, class A, C, and D are based on serine residue at the active site of the beta-lactamases. With class B, these are essentially based on using zinc to break the amide bind. So these are called metallo-beta-lactamases, and these are perhaps the most resistant organisms when they have metallo-beta-lactamases. So some examples are IMP, WIM, and NDM. Class C, which is essentially AmpC for the most part, and there are other enzymes in this class, was the most clinically relevant is AmpC, and these essentially break down cephalosporins, but they can also break down penicillins and penicillins. And these AmpCs, they are stable to cefepime, so it cannot break down cefepime, but also they are stable to carbapenems, so they cannot break down any carbapenems, including imipenem, meropenem, doripenem, and ertapenem. And lastly, we have class D, which can include ESBL. So there are some OXA type ESBLs, but also most of these are carbapenemases. So a lot of, there are various numbers of OXA enzymes, and most of them are carbapenemases. And, they break down penicillins, cephalosporins, as well as carbapenems. And of course, because we have all these beta-lactamases, we have also developed beta-lactamase inhibitors. So we have several beta-lactamase inhibitors on the market. So classically, we have clavulanic acid, which is actually a natural product. And it can essentially inhibit TEM enzymes, SHEV enzymes, and CTX-M. Has no activity against class B, C, or D beta-lactamases. Then we have sulbactam, which is not the best for ESBL, so it doesn't have good activity against CTX-M. It has some activity against TEM and SHEV. Does not have activity against class B, C, or D. But most importantly about sulbactam, it has, unrelated to beta-lactamases, it has intrinsic activity as an antibiotic against Acinetobacter baumannii. Tazobactam also can inhibit ESBLs, including CTX-M, but no activity against class B, C, and D. Enmetazobactam is a derivative of tazobactam, so it has improved potency against ESBLs. That's what it's designed for, so better activity against CTX-M ESBLs. No activity against class B or C, and there's limited data for class D. It may have some activity against some oxas, but right now there is limited data. Then we have avibactam, which has really good activity against class A, including ESBLs as well as KPC. So these are, carbapenemases. So avibactam can restore activity against carbapenem-resistant Enterobacterialis. Does not have activity against class B, but also stable against class C, AmpC, and OXA48. So OXA48 is inhibited by avibactam. Relibactam, also good activity against ESBLs and KPC, as well as AmpC, and limited activity against OXA class D enzymes. Durlobactam, also excellent activity against ESBLs and KPC, AmpC, and a large variety of OXA enzymes in class D. Weberbactam, great activity against ESBLs and KPC and AmpC. And lastly, taniborbactam, which as of this recording still under development. It is not on the market yet. It has activity against ESBLs and KPC. It has activity against some class B. So this is the first time we have a beta-lactamase inhibitors that may inhibit some of the common class B. And that's not all of class B. So there is, for example, IMP that's not inhibited by this, but certainly NDM and VIM are. It also inhibits AmpC and has activity against OXA48. Now let's take a look at these beta-lactamases in another way. So essentially beta-lactamases, you can think of them as serine beta-lactamases or metallo-beta-lactamases. Metallo-beta-lactamase, which is the worst type of beta-lactamases. These are amyloid class B. These are carbapenemases that break down carbapenem, cephalosporins, and penicillins. Some examples include IMP, NDM, and VIM. And we don't really have anything on the market as far as beta-lactamase inhibitors. Taniborbactam is under development that could inhibit some of these. For serine beta-lactamases, we have amyloid class A, which includes the narrow spectrum like TEM1 and SHEV1. These are the ones that inhibit penicillins and first or second generation cephalosporins, and they're essentially inhibited by all beta-lactamase inhibitors that we have. Then we have ESBLs, and most commonly is CTX-M. These are inhibited by most of beta-lactamases we have except sulbactam. And then the worst type of class A is carbapenemases, so KPC is the most common one. and that's inhibited by some of the broad spectrum beta-lactamase inhibitors, namely avibactam, relibactam, durlobactam, vaborbactam, and taniborbactam. With class C, which these are cephalosporinases that also break down penicillins. These are inhibited by avibactam, relibactam, durlobactam, vaborbactam, and taniborbactam. And lastly, we have class D, which are carbapenemases, and these are oxa types. That includes some ESBLs, but most of them are carbapenemases. So carbapenemases, they will break down penicillins, cephalosporins, as well as carbapenems. Most common one is OXA48, and OXA48 specifically is inhibited by avibactam and taniborbactam, and durlobactam is unique in the sense that it can inhibit most of OXA type enzymes. Let's take a look at Staphylococcus aureus. So Staphylococcus aureus can be divided based on its pattern of resistance. So the wild type, which is basically penicillin susceptible, is constituting less than 10% of the isolates nowadays. Then we have MSSA, which is methicillin susceptible. MSSA is resistant to penicillin. Then we have methicillin resistant, so MRSA, and the drug of choice for MRSA is vancomycin. Now, while very rare in the United States, there is also vancomycin intermediate Staphylococcus aureus, or VISA, and that is basically that Staph aureus is no longer susceptible to vancomycin, but the MICs happen to be in the intermediate range. And then we have Staphylococcus aureus with MICs in the resistant range for vancomycin, which is referred to as VRSA. Now, MRSA is clinically important, and in general, there are different types of it. So we have healthcare associated MRSA, and then we have community associated MRSA. Now there's also livestock associated MRSA based on the antibiotics that are used in animals and in food production. But clinically we basically break these down into healthcare associated MRSA or community associated MRSA. And healthcare associated is the more common strain which most healthcare professionals encounter. And this strain is resistant to beta-lactams, with the exception of ceftaroline, and several other classes, making prefer therapy more broad spectrum agents. Now, community-acquired MRSA is strain only found in the community, and other classes other than beta-lactams are still possibly active against these, so such as like doxycycline, moxifloxacin, trimethoprim sulfamethoxazole. Now, something of note is that the resistance genes that make Staphylococcus aureus resistant to methicillin is the MECA gene, which can be identified on rapid diagnostics. And this is the gene responsible for alterations in penicillin binding protein 2A, and that is the target site for methicillin class. So nafcillin and oxacillin, as well as cefazolin, they bind to the site. So when there is mutation, alteration in this, they no longer can bind to penicillin binding 2A, which makes it resistant to that class. MRSA is also considered a serious threat by the CDC, but the good news is that it seems that the number of cases year after year are declining, and in recent time it may have stabilized. The issue is that MRSA has become resistant to many first-line antibiotics, specifically oral antibiotics that we may have for less serious infections. And people who inject drugs are 16 times more likely to develop a serious MRSA infection than those who do not. Enterococcus species, which are gram-positive, can develop resistance to aminoglycoside in 25 to 50% of the isolates. And these are basically plasmid-mediated resistance that are aminoglycoside-modifying enzymes. And of course, there is vancomycin-resistant enterococcus, or VRE, and there are two main genes that make VRE, so VanA and VanB, both of which can be identified with rapid diagnostic tests. And basically, the site of action for vancomycin is the alanine dealanine, and what happens in enterococcus with these genes is that basically it mutates and changes the second alanine to a lactate, and as a result, vancomycin can no longer bind to the alanyl D-lactate, and this mutation basically does not interrupt the life cycle of Enterococcus. So Enterococcus at this point will be VRE, it continues to replicate, and vancomycin will be ineffective. Now, Enterococcus is part of the human flora, so it is common for people to be colonized with VRE, and that's something that can be detected by screening tools on the stool sample. Vancomycin-resistant enterococci, or VRE, are also considered a serious threat by the CDC. You can see that year after year, the number of cases have been going up. However, in recent time, the number of cases may be going back up again. And about 30% of all healthcare-associated enterococcal infections are VRE, and nearly all VRE infections happen in patients with healthcare exposures as opposed to happening in the community setting. VRE is increasingly resistant to additional antibiotics, raising concern that the remaining drugs to treat VRE may become less effective. Lastly, let's take a look at E. coli. So E. coli is a gram-negative rod, and it can be resistant to many antibiotics, so including resistant to beta-lactams, and it's primarily through various beta-lactamase production. So including some strains can be ESBL producing, as well as KPC, which is a carbapenemase, as well as OXA48, which is a carbapenemase, and of course the whole subclass of metallo-beta-lactamases that are carbapenemases. So E. coli can be very resistant. At the same time, a wild-type E. coli can be pan-sensitive to many antibiotics. Now, trimethoprim sulfamethoxazole resistance is basically increasing, and it is due to overproduction of dihydrofolate reductase. So dihydrofolate reductase is the site of action for trimethoprim, as well as mutations in the gene DHPS is the site of action for sulfonamide, so sulfamethoxazole. Fluoroquinolone resistance can be due to polymorphisms in the GIR-A, which is a gyrase, the gene for the gyrase, and basically that's the site of action for fluoroquinolone, such as ciprofloxacin. Tetracycline resistance can be induced by expression of Efflux pump, which is encoded on the TET genes.